This program is brought to you by Stanford University. Please visit us at stanford.edu.
Good evening, everyone. My name is Phil Pizzo. I'm the Dean of the School of Medicine here at Stanford, and we're very, very pleased to have you with us this evening for what is now the fifth and last of these wonderful summer sessions. And I hope many of you have had a chance to enjoy prior ones, but I'm quite sure if you haven't, you'll enjoy this one for sure. I want to tell you that this particular event this evening is being videotaped, so that you're aware of that. And we will have an opportunity during the discussion, during the presentations for discussions. We would love this to be a really interactive forum. We're going to be discussing tonight what is arguably one of the most important, albeit also one of the most controversial areas of modern medicine. When I think back over my own career during the last 30 to 40 years in medicine and science and reflect on what has changed, it has been certainly remarkable. But my experience in the area of stem cell research, particularly since I've been here at Stanford and in California, has given a new meaning to that. Probably because this is also a point in our history when we're witnessing the convergence and divergence of views regarding science, religion, ethics, and various moral points of view. And in fact, a lot of anti-science thinking as well. You know, it's a time when, even in this country, some 40% of Americans don't believe in evolution, and many have very strong views about the validity of conducting stem cell research. And while I certainly respect everyone's point of view, I also believe that this is certainly one of the most promising areas of current investigation that has truly an opportunity to transform the way we think about the delivery of care in the future. And of course, if we don't proceed, we certainly have the risk for losing our edge in this country. There was a time almost 25 years ago when a similar issue arose over the whole theme of genetic engineering, and one of the most famous scientists here at Stanford, Dr. Paul Berg, really helped to shape the agenda that led to genetic engineering, and it almost too came to a halt. But then, thankfully, reasoned minds prevailed, the science went forward, and the whole evolution of biotechnology was a consequence, and many drugs and treatments that we have today are available because of that work. And one must envision that if we don't proceed with stem cell research here in this country, it will certainly transform the world because it will take place elsewhere, and witnessing that happening right now. I've had the opportunity during the last 2 years to serve on the oversight board, the so-called Independent Citizens Oversight Committee, for what was once known as Prop 71. Perhaps a number of you voted for Prop 71. We know 60% of Californians voted for it, so at least I could say probably 60% of this group did, maybe 100%. That would even be better. And I must say to you that on November 4th, when that happened, I felt really proud to be a Californian. I came here from Boston just a handful of years ago, but I was pleased that day because it meant that citizens were speaking up and about an issue that we felt very passionate about. Of course, 5 years ago, in August, President Bush made his decree regarding federal funding for stem cell research, and so in some ways Prop 71 was an event or a consequence of that. It was an opportunity for states to say, if the federal government, which has invested so much in biomedical research, is not going to do that, we need to, such as in California, do something about that. And I must also tell you that we and my colleagues on the so-called ICOC that oversees the California Institute for Regenerative Medicine have really spent countless hours during now almost the last 2 years working to get the systems in place so that this Institute for Regenerative Medicine could make a big difference here in California. And while we've gotten everything in place, until just a couple of weeks ago, we really had no funding because the litigation that came forward from a couple of groups blocked our ability to bring funding to Prop 71. So at this juncture, it really has remained largely a promissory note. But we believe, thankfully, that that will change in the months ahead, and I think with that, we'll have an evolution of new insights and new science, and eventually, hopefully, as a consequence of that, new therapies. Now, tonight we have two wonderful speakers who are going to reflect different portions of the issue of stem cell research with you. First is Julie Baker, who's a professor in genetics, and she's going to talk to you about what stem cells are and help you better understand some of the controversies and issues that surround the science. You know, one of the unfortunate things is our science has been confounded by words that confuse the public, confuse all of us. Things like cloning, things like that, which are very different from what one is thinking about stem cell research. So the first part of this is to understand what it really means. And then Dr. Hank Greely, the Johnson professor of law, who also has an appointment in genetics in the School of Medicine, is going to speak with you about some of the legal and ethical issues that surround this important topic. And then we'll have an opportunity for questions and answers. So we'll have about 15 to 20 minutes of presentation by each of our two speakers, and we'll try and save the bulk of the time until the sun sets to really have an opportunity for dialogue. And hopefully at sunrise tomorrow, we'll all be ready to move forward. So I'm going to begin by having Julie Baker come forward to begin this presentation.
I'm going to put some of my props out here from the get-go. Thank you, Dean Pizzo, and I just wanted to mention that he called me a professor of genetics. I'm actually an assistant professor of genetics. That means my tenure package will be on his desk in about a year, so I'm just letting you know that. Thank you. I'll have that in writing. Actually, I think it's being videotaped, so good. That's good. We've already accomplished something. So first, I just want to thank you very much for coming to this. Tonight, I'm going to talk about what a human embryonic stem cell is. And as I was preparing this talk, I really thought, you know, this is actually the most important lecture that I've ever given in my scientific career. And that's because it's actually, at this time in our history, it's actually imperative that our communities really, really understand these issues. And I've heard there are teachers out there. I hope that there's, after tonight, that you can go back to your classrooms and tell your students exactly what's up and exactly what the issues are, as well as non-teachers. You can talk to your neighbors, your friends, your family members. This is incredibly important, and it's just so nice for me to be able to convey this message to you all tonight. So I really have four main issues that I'm going to talk about tonight. The first is I really want to nail home the idea of what is regenerative medicine. Okay. The second is what is a human embryonic stem cell. The third is where do embryonic stem cells, human embryonic stem cells, come from? And the last is to convey to all of you that the time to do research on human embryonic stem cells was actually yesterday, but with what we've got, we need to do it now. Okay, so those are the first 4 points I'm going to discuss tonight. So I'm going to start with what is regenerative medicine. Regenerative medicine is the concept that in the laboratory we can actually engineer tissues, and that we can then give those tissues to the clinicians who can put them back into diseased organ systems and cure and treat disease. So an example of this might be in diabetes. So diabetes is an epidemic, especially because obesity is occurring at astronomical rates in our country. And so the idea would be that as a scientist, we make pancreas cells in culture in the laboratory, and then use those pancreas cells to put them back into a patient that has diabetes and cure the diabetes. So that's sort of the general idea. And this sounds like rocket science, right? It sounds like some futuristic ideal. But I'm here to say that it's not, in that types of regenerative medicine have been practiced in the hospitals for many years now. And I think the most important example of this is in bone marrow transplants that are used to cure diseases of the blood. So in those circumstances, a patient with leukemia, for example, comes into the clinic, and drugs are used to get rid of their blood system and therefore get rid of the disease. And a bone marrow transplant is used then to replenish the blood in that patient. So you've gotten rid of the disease, and now you've replenished the blood system from a donor tissue. The reason that we can do this is because the blood is very special and that it has a blood stem cell. So this is an adult blood stem cell that resides in everybody's body. This is why you can donate blood and a week later have it all back. You've got these stem cells that are making all the cells in the blood and replenishing your supply. So that's great with the blood, right? What an amazing organ the blood is. Or I guess it's not an organ, but. So what about the brain? What about the spinal cord? What about the pancreas, the heart, the lung? Can we do that with those tissues? No. There are stem cells in some of those tissues, but certainly not all of them. And the stem cells that exist don't do the same kind of knockdown, drag-out job as the blood stem cells. For example, the stem cells in the brain can help they can help heal stroke. They don't do, they don't completely rescue the problem. So we need to turn to the embryo. And I'm here today to tell you that we need to turn to the embryo. And that brings me to the topic of human embryonic stem cells. The embryo is perhaps the most miraculous thing in nature, in that it starts as a one-celled embryo and can become any tissue, obviously, in the body. You and I were all testaments to that. So this brings me to the idea of the human embryonic stem cell. So what is a human embryonic stem cell? A human embryonic stem cell comes from a human embryo. And the miraculous thing about a human embryonic stem cell is that if you push it in the laboratory using different drugs, using different conditions, that cell can become any tissue in your body. It can become liver, it can become lung, it can become a neuron. And I have one prop to start with. Human embryonic stem cell lives in a dish like this in the laboratory. And several million of them live on a dish like this in the laboratory. So they're very, very, very small. And you can only see them under a microscope. Okay, that's what they are. And if you push them in different directions in the laboratory, we can actually see neurons. We can see heart muscle. We can see all the tissues of the body on dishes like this in the laboratory. And I actually brought a movie that I'd like any of you to come up after the talk to see. And it's a movie that was created in my laboratory on human embryonic stem cells that we in the lab have made into heart cells, okay? And these heart cells will form balls, form blood inside the balls, and the heart, this synthetic heart starts pumping. And you can actually see that in the laboratory. Dishes like this full of cells that are pumping. It doesn't take a lot. It doesn't take any sophistication to understand what that might mean to regenerative medicine. That we might be able to take those cells and put them back into diseased hearts and cure the problem. And there are actually researchers at Stanford that are working on that problem. So that's an embryonic stem cell. That's the power of embryonic stem cells. And I'd encourage you to come look at the movie after this is all over. Okay. So one of the big problems today is this issue. Where does a human embryonic stem cell come from? Okay. So in your mind right now, I want you to close your eyes. What does that embryo look like? Given the fact that a human embryonic stem cell comes from a glorious human embryo, what does it look like in your mind? I actually presented that to a class that I was teaching of graduate students at Stanford, and I had them draw on a piece of paper what they thought that embryo looked like. And most of, I would say 95% of the papers that I received back were little babies with an umbilical cord to the mom. And, I'm sure this is a much more sophisticated audience. So let me tell you what these embryos look like. So first, I'm going to present human embryology 101. Okay, so we have an oocyte, an egg, and a sperm from the father. They meet. We get a one-cell fertilized embryo. The next day, 24 hours after conception, that embryo divides into 2, then 4 cells, then 8 cells, then 16 cells. Now we're at 4 days, and we have something that's called a morula. That's just jargon, but it's a 32-cell embryo. And what happens at that stage is the embryo squishes down, becomes a really compact ball of cells, and then it explodes the next day into a giant ball. And that's what my little demonstration is here. This is a human embryo that's been magnified several million times. And this is what it looks like. And I want to emphasize to you that this is free-floating, so we can keep this in culture in the laboratory. It doesn't need a uterus, doesn't need a blood supply or nutrients from the mom, and it's about 4 or 5 days after conception. Okay, this is what it looks like. It's a hollow ball. It has these cells on the outside, which in this model are depicted as pink and blue and green and white, or see-through or something. Those cells are the placenta. They're going to become the placenta, these guys. Okay, so most of the cell is actually thinking about becoming the placenta, because in the mammal, that is the key, right? You got to have the mom from an early time, and maybe even years and years after, as I found out. And then down here at the little teeny weeny corner in the bottom, there's this little cluster of cells. There's probably 16 or 20 of them, and I've drawn them on this ball. Little black cells. Those guys are the embryo. That's the embryo right there. And the miraculous thing about these 16, 20 cells, whatever it is, it depends on the embryo, is that we were all that. Every single last cell in yours and my body came from those 16 cells. Every last one of them. Okay, so that's what we're dealing with, these guys. And a human embryonic stem cell, basically what happens is that this embryo is plated, put down on these dishes, and the placental cells die, and these cells take over the dish. They can self-renew, so they can keep propagating themselves pretty much indefinitely. They're sort of immortal. Okay, so that's a human embryonic stem cell. That's where it comes from. Okay, now the big question, the one you've all been waiting for, is where do these embryos come from? Okay, and for a minute I want you to imagine that you're IVF doctors. Okay, and that you're looking through a microscope at a cohort of embryos that resulted from a couple that couldn't have a child of their own. So that is where the embryos come from. They come from in vitro fertilization clinics where patients come who want children. Okay. So this is the microscope stage. You all are like looking down on it, and I'll hold them up for the guys, all of you back there. Okay, so your patient, you come into in vitro fertilization clinic because you want a baby. Okay. And the average IVF cycle yields about 10 to 15 embryos. And these embryos are not equal, right? So of the 10 or so embryos, you get all kinds of stuff. You get really, really, really healthy embryos, and you get really strange-looking embryos, and you get everything in between. Some develop and culture nicely, others don't. Okay. So as a doctor, you're sitting there looking at the microscope at this embryo picture, okay? So here we have 2 embryos. They look really, really, really good, right? They're huge. They've expanded. They have this cluster of cells, and their placental cells look great, which is necessary for binding to the uterus. Then you have kind of intermediate ones. You know, they've expanded. They're kind of big. They have both cell populations. You know, maybe they'd become babies. Then you have some that, oh man, you know, these 3 just, they don't look that good for various reasons. This one's arrested at 4-cell stage. This one just looks strange. Maybe this one, the placental cells aren't even there, so you're never going to get a child if you don't have placenta. Just never going to happen. And this one just, you know, died a long time ago. My father didn't have the energy to blow up the 3rd one. Okay. So for a minute, I want 3 volunteers. Can I just get quick? Maybe somebody would help me here. Thank you. Okay, you're the freezer. You're discard. Aww. And you're the mom. Okay, so now I'm the doctor, right? So I'm going to say, okay, you know, I'm taking these 2. These 2 have the best likelihood of giving this mother a child. And it's now considered ethical to put back only 2 back into the mom. At one time, they were putting 10 back or whatever. It's now and that is no good for anybody. So it's now ethical to pick 2, and you get these. Okay, Mom. Okay, so okay, and so you know, I think these are kind of good. I'm thinking these are good. They're just not big. So, you know, that doesn't always matter. So when I'm thinking about this, I think we'll freeze these. Because if she doesn't get pregnant, she can always come back, and we can thaw these out and try again. No use putting her through all the hormones and everything necessary. So we're going to put these back in the freezer, so you get these. Okay. And what am I going to do with these? Any ideas? We're going to discard these, which is a really nice way of we're throwing them away. Okay, so you get these. That is what happens at the IVF clinics, right? That's exactly what happens. And in my lab here at Stanford, we are interested in the development of new human embryonic stem cell lines. And we've derived 4 new human embryonic stem cell lines in my lab. And Where did those lines come from? 2 of them, exactly, came from the discard pile. Those would have been thrown away. We used them. We got 2 lines, healthy lines that can do all that, pushing them to heart muscle beating, pushing them to neurons. Great. And the other 2 came from the freezer. Okay? So that's where they came from. Let me just touch point on the freezer for 1 second. In that there are an estimated 400,000 of these embryos in the freezers in the United States today. 400,000 of them. When we thaw these embryos out, we lose 50% of them. 50% of them. So now we potentially have 200,000 viable embryos. To date, 84 of these embryos have been used to generate babies. That's it's a big difference. Okay. Those are Bush's snowflake babies. We got a long way to go. It's a lot of kids. Okay, so just thought questions for a second, and then I'll let you guys go. What are we going to do with all those embryos? Okay. Does anybody want to volunteer to adopt them? And secondly, why can't we use the discarded pile? Okay. Thank you. Thank you very much, you guys. Okay. So the question I always get is, why do we need to derive new human lines? Am I back on? Why do we need to derive new human embryonic stem cell lines? In August 2001, President Bush said that after this point in time, there will be no federal funds used for the experiments done on anything that hasn't been created yet to date. And that left us with a so-called 60 human embryonic stem cell lines that the government would approve for us to use. What I mean by a line, and I should clarify this, is that each embryo that develops into a stem cell, that's a line. So each line comes from one single embryo. We don't mix up the embryos, okay? So there are 60 lines. So after that date, a bunch of us across the United States started looking at those 60 lines that we could use. Turns out that there's probably only 5. There's probably only 5. And I would say 99% of the research on human embryonic stem cells in the United States are done on 2. And most of those are actually done on 1. Okay, so, we need more. The second reason why we need more is all of those lines, all 5 of the usable ones, were generated, were experimented on, were created in animal contaminated conditions. So, in any of the cultures of those lines, there's products from the pig, products from the cow, products from the mouse. Okay. That's never going to cut the mustard when you're talking about FDA approval to actually do transplantation. Never will work. Okay. The second is that those lines are growing old. So, and they're accumulating rapidly chromosomal and genetic abnormalities. That's multiple papers have been published on this. Okay. And the fourth line, which is something that we're really interested in pushing forward at Stanford, is to generate disease-specific human embryonic stem cell lines. And there's a lot of jargon there, but what that means is that we'll take an embryo that the IVF clinic has said, if this embryo grows up, it's going to have cystic fibrosis, or it's going to have multiple sclerosis, or it's going to have Parkinson's disease. And the patients want a normal embryo. They want a normal child. They'll put back the normal children, and we'll get these. And the value of these lines is that they are tremendous models so that we can study those diseases. We can put drugs on them to see how neurons in Parkinson's disease will react to a whole battery of drugs. We can study the mechanisms of action of how the neurons in Parkinson's work because we can make them. And we don't have to do this in patients. Okay, this is a huge deal and one of the major emphases here at Stanford in my lab. Okay, so with that, I'm just going to have a few closing comments. So I'm a junior professor here at Stanford. And everybody always says to me, why are you doing this? This is a huge bureaucratic nightmare. And you've gotten yourself into it. And the reason is because I really believe that these cells hold the key to a variety of future therapies that will help treat a large array of human diseases. And that progress across the country has definitely been hugely hampered by the federal restrictions. So what I want to convey to you today is that it's going to take lots of time, lots of scientific energy, lots of patience from the community, and a lot of money. But I think that these cells are powerful, and I think that we're going to get there. And with that, just thank you.
So we're going to move now to a review by Professor Hank Greely on some of the ethical and legal issues, and then when he's done, we'll come back and have a broad discussion about this topic. As you can see, he's rounded out, so he is a full professor.
Thanks, Phil. Well, good evening to you all. I want to first start by complaining that it's unfair for me to have to follow Julie. I don't have any clever visual aids, and I'm not young and brilliant and good-looking and all those other things. And in fact, I'm even a lawyer. And for about the last 15 years, my work has really focused on legal, ethical, and social issues in a variety of biosciences, genetics, neuroscience, stem cells, cloning, a bunch of different things. And I find them all fascinating. But I have to confess, most people don't find them as fascinating as I do. And I think there's a reason that all of you are out here to hear about human embryonic stem cells, and you wouldn't necessarily be out here to hear about confidentiality protection and phenotype-genotype resources. Julie's talk gave you part of that reason. But Julie herself is another part of the reason. Julie is a mother of a 3-year-old and a 3-month-old. While she was raising the cell line, she was giving birth to another daughter. Julie is a brilliant scientist and obviously a great teacher. Julie also, in the state of South Dakota and in the country of Italy, would be a criminal and might be behind bars. More likely, she would have left South Dakota or Italy to come to a place like California. Unlike most areas of science, this is extraordinarily contentious, including contentious to the point of involving the criminal law. And that, I think, is why not just I'm fascinated, but why you people are here to hear in part about the legal and ethical implications of it. I have to confess, though, that I'm here wearing kind of two awkward hats. I am an academic. I'm interested in intellectually understanding these legal and ethical implications. But I've also been sucked in to become part of that massive bureaucracy that Julie complained about. I think that I'm on 4 different Stanford committees that help try to make sure that Stanford uses human embryonic stem cells in ways that are both legal and ethical. I chair a California committee that's coming up with recommendations for how California should regulate human embryonic stem cell research. So part of me is the academic, a disinterested academic, and part of me is the regulator, bureaucrat, advisor, trying to deal with practical problems and come up with solutions to them. Those two parts live uneasily inside the same skull. So as you listen to me, remember that I'm kind of a chimeric creature, part bureaucrat on this issue, part academic. What I want to do in the short time I have this evening is talk about three things. First, I want to talk about the issue of the moral status of the embryo and the implications that has for this debate. Second, I want to talk about a handful of the many other really difficult and important practical, legal, and ethical problems that we face if we decide to go forward with this kind of research. And then I want to end with a few thoughts about what this debate tells us, what this controversy tells us about the relationship between science, politics, and society. So let me start with the question of the moral status of the embryo, or as my Stanford colleague David Magnus likes to call it, the question of what's in the dish. Some people think what's in that dish that Julie showed you is a human being, entitled maybe not to drive or to drink or to vote, except maybe in Chicago. Now, if you can vote while you're dead, why not? But entitled to life, liberty, and the pursuit of happiness, and at the very least entitled not to be murdered. That's one extreme of the discussion. The other extreme says that's just a clump of waste human cells. It's like a tumor biopsy, a tissue biopsy, or a little tag of skin that you pull off. It's nothing. It's waste product. We don't have to worry about it at all. Now, I've talked to a lot of people about this issue over the last several years, and I don't really think I found anybody who's truly at one of those extremes or the other. I found some people who say they are or who think they are, but if you push them on it, they really aren't. So let's take the equal human being extreme. If you find someone who takes that position, ask them. Give them a hypothetical. We law professors love to give hypotheticals. You are in an in vitro fertilization clinic. A fire breaks out. There is one 4-month-old baby you can save, or you can pick up a tank with 1,000 frozen embryos. Do you save the 1,000 equal human beings, or do you save the one baby? And I've yet to find anybody who goes for the tank. On the other hand, if you really believe this is just a bit of cells, just a bit of tissue and waste, well, I ask, so okay, let's say we can turn human embryonic stem cells into a great production line for making lawn fertilizer or cat food. And nobody seems to like that either. So I don't think anybody's truly at those extremes. We're all someplace in between. And many of us are in different places in between. Personally, I've come to figure out where I am, but I think where we all sit depends in part from our perspective on what those embryos, how those embryos came to be, how they came into existence, where, if anywhere, they're going. Are they going in the discard pile, the freezer, the mother? And most importantly for many people, what are they being used for? Are they being used for a good and important purpose? And many of the people who support this research say, saving the lives or improving the quality of lives of living people who've got terrible diseases is a plenty good purpose for sacrificing some embryos that weren't going anywhere. Other people take the other position and say, well, you'd slice up babies to save a life, and that would be wrong. Now, personally, I am inclined more on the use the embryos position, and part of it is because those embryos, unless they are implanted successfully in a woman's womb, in a properly prepared, properly timed woman's womb, they're not going anywhere. And even if they're implanted in a properly primed womb, they only have a 1 in 6 chance of becoming a pregnancy at all. And on top of that, through the natural, old-fashioned way of making embryos, which we call sexual intercourse, anywhere between 50 and 80% of the embryos that are created every night and occasionally during the day throughout the world never become fetuses, let alone babies. 50 to 80%. Now, there are about 5 million pregnancies a year in the United States. If 50% of the fertilized eggs never become fetuses, that means 5 million embryos die each year. If 80% don't become fetuses, That means 20 million embryos die every year. Well, we lose about 2.5 million people a year to death in the United States. If you really think that an embryo is a full human being, and its death is the same as the death of a 90-year-old or a 60-year-old or a 13-year-old, that's an awful lot more deaths, and we don't act like it. Now, it's true we don't always know when it happens, but we don't force people to take enormous precautions to preserve those lives. So to me, that's kind of some evidence that we don't really think of them as lives. But one thing I've definitely discovered over my years of talking about this is some people hear that and agree with it. Some people don't agree with it. I'm never able to convince the people, or almost never able to convince people who disagree with me that I'm right. And they're never able to convince me that they're right. This is an issue where people have strong feelings and strong beliefs, and they're not moving very much one way or the other. As a result of that, this what's in a dish problem turns out to be quite significant. If you think this is really important research and there's nothing wrong with doing it, you favor going full speed ahead. If you think it's killing babies, you don't. Every jurisdiction in the world with significant biomedical research has now come up with an answer, its own answer, to what's in the dish. South Dakota and Italy say it's a baby and it's a crime to kill it. In the UK, you can use these embryos in various purposes, but only with a license from a governmental authority. In Germany, you can't make German embryos and German embryonic stem cells, but you can use non-German ones. Which isn't quite fair to the Germans, but it isn't quite unfair either. You can't make cell lines in Germany, but you can use cell lines in Germany that have been made by somebody else, somewhere else. In France and in Canada, you can make embryonic stem cells from the freezer, from the frozen embryos, but you can't take fresh embryos and make them. Most American states, well, in the federal government, there's nothing illegal about human embryonic stem cell research, but you can't use federal funding either to destroy an embryo or to do research with embryonic stem cells that were derived after August 9th, 2001. Most American states have answered this question by not answering it. They don't have any law on it one way or the other. There are about 15 that do. So in those states, it's legal, but it's not particularly encouraged. And then there's some states, including California, New Jersey, Massachusetts, Connecticut, and a few others, Illinois, that have said, we like this research, we encourage it, it's legal, in fact, we'll give money for it. And now, finally, some of that money is beginning to flow. So in the United States and around the world, different places have answered this question different ways. I think that's okay. I'm a Democrat with a small d, also with a big D, but that's irrelevant right now. If a country doesn't want this, then it's their right to choose not to have it. If South Dakota doesn't want to do this research, that's fine. I hope South Dakotans don't get first in line for any cures if the research leads to cures. But you know, we won't begrudge them. We'll be so happy if the research leads to cures that we'll share it with everybody. So in the democratic process in various places, different places have reached different results. Those results aren't necessarily permanent. They may change. A legislature may change its composition. People may change their minds. A presidency may change. These rules may change one way or the other. But I think one of the key things that is most important in whether those are going to change or not is the alternatives. Because one argument that I think has some force is, if we can do this in some other ethically less problematic way and get all these benefits, let's do it that way. If there's an ethically pure and clean way to do it, let's use that. And people have suggested some alternatives. The use of the adult stem cells that both Phil Pizzo and Julie Baker mentioned, the use of umbilical cord stem cells, the use of sort of embryonic stem cells made from embryos that aren't going to give rise to children, not because they won't be implanted, but because for some physical reason they can't. So trying to get live cells from dead embryos. Whatever it means to say there are live cells in a dead 8-cell embryo, it's not clear how you would define that, but that's one proposal. Our Stanford colleague Bill Hurlbut has recommended something called alternative nuclear transfer, where you basically use the the cloning technique, the somatic cell nuclear transfer technique, to take an embryo and make sure it can never become a baby, and then use those cells. There's a proposal to use sort of embryos made through parthenogenesis, which is where an egg starts acting like it's been fertilized when it hasn't been. Now, if you're a snake or a lizard, you can actually have live births that way. Mammals don't. And then finally, there's a proposal to take single cells off of these 8-cell embryos and turn those into embryonic stem cell lines while saving the 7-cell embryo. That's what you may have read about this past week in the news. Advanced Cell Therapy, Advanced Cell Technologies has announced and published in Nature that it can do this. It was a little bit tricky. They said they could do it. It wasn't exactly what they did do. They actually destroyed the embryos they were working with, but I think they're right that in principle they've showed that you should be able to make it work by just taking one cell off. Now, all of these alternatives raise two important questions. How plausible is it that this will deliver on the scientific promise of human embryonic stem cells? And how ethically problematic is this alternative? With respect to the scientific plausibility, right now the gold standard is the embryonic stem cell, because we know it can form each and every one of the 300 cell types in our body. It can make brain neurons, it can make skin cells, it can make liver cells, and we know that to an absolute certainty because it did it for each one of us. All of our cells, all of our cell types started out as embryonic stem cells, as that inner cell mass in those blastocysts. So we know that works. There is controversy over whether adult stem cells could be turned into other cell types, whether umbilical cord cells can. Some say they can turn them into some other things, others say we can't do it. With respect to the other alternatives, we just don't know yet, including this new ACT method. We never believe anything in science until it's been replicated, usually 2 or 3 times, and that's, of course, after the Korean scandal in embryonic stem cell research, particularly important in this area. I'm not accusing ACT of fraud, but science, sometimes one lab does it and nobody else can figure out how to do it. And the first lab may not be able to do it again. So we don't know whether they can do it reliably and efficiently, and we don't know whether those cells will be the same and as effective as human embryonic stem cells. But that's probably the most plausible of the alternatives. But then you've got to ask, is it ethically plausible? Will it satisfy opponents? And there, at least the verdict is in from some of the opponents who have already said this ACT method is just as bad as the first method. You're not saving an embryo, you're twinning. You're turning one embryo into 2 people, and you're killing one of them. And if that's your perspective on it, then it's not an ethical advance. So the alternatives are interesting. I think from my perspective and the perspective of the scientists I deal with, the answer isn't let's do human embryonic stem cells and nothing else. It's let's do them all. And if some turn out to be scientifically and medically as useful as we hope, focus on those. And if we get 2 alternatives and one is ethically better than the other, focus on the ethically better one. But we are so far right now from knowing what's going to work medically and ethically that it's way premature to make those decisions. So, second big point. Most of the interest, most of the focus in the ethical debate has been on this what's in the dish question. And perhaps not unusually in ethical debates, for all the billions of words that have been spilled, I don't think very many minds have been changed one way or the other. But while that's been going on in places that have decided they can do this research, places like California, places like Stanford, we have to decide a host of other issues, practical issues about doing the research. There are, 10 or 12 at least, but I want to mention briefly 4 of them. The first deals with getting eggs. Now, Julie's stem cell lines and the form of making stem cells Julie told you about takes leftover embryos from in vitro fertilization clinics. But there are scientists who want to, and California law encourages this, want to make new embryos, taking an egg and fertilizing it with a sperm to make a new embryo to be used for research purposes. But then that raises the question, where do you get those eggs? The embryos are lying in these freezers, as Julie pointed out, and they're created in huge numbers every day in IVF clinics. But except we're just now beginning to freeze eggs, but there's not a whole bunch of frozen eggs out there. Eggs are fresh. When you get eggs, they're from somebody who's gone through an egg harvest procedure and is usually about to want to use them for in vitro fertilization. What do you ask the woman who's had 10 eggs taken from her and wants them to use them to make a baby? Can we take 2 of these 10 and use them for research? That's a pretty hard thing for her to say when she doesn't have a baby yet. Or you can take donors, which is what's done in vitro fertilization for egg donation. You have to put, in this context, donation in quotes, because you may, well, take a look at the Stanford Daily, the online or the paper edition, and you'll see ads for $10,000, $20,000, $50,000. The biggest I saw was $100,000 for a woman of South Asian descent who had to be a certain height, certain SAT scores, and athletic. $100,000 for an egg donation. If you're getting $100,000 for it, donation doesn't strike me as quite the right term. But in fact, women typically get at least $5,000 for it, and that's not at all implausible because donating eggs is not an easy matter. You get shot full of various drugs for a month or 2 beforehand. They bloat you and make you uncomfortable. You then go through an uncomfortable surgical procedure. There's some after effects. 1% of the women who go through this end up hospitalized for some time. And although it's rare, occasionally a woman has some long-term disability, reproductive or otherwise, and there's at least one report of a woman dying as some of the side effects of this. So it's not a trivial thing. Sperm donation, trivial thing. And the pay is much lower. But egg donation is not a trivial thing. Well, if you want to get eggs for research, should we pay people or not? And can we get eggs for research if we don't pay people when the in vitro fertilization clinics are paying people $5,000, $10,000, $20,000 to go through it? How many people will do it for free? Some, perhaps, but so far, at least, apparently not very many. So one ethical question is, how do you get the eggs? How do you ethically get the consent? When do you ask a woman who's going through this? Do you pay her or not? And there's a bill that has just been passed by the California legislature that's sitting on Schwarzenegger's desk to be signed. I think he'll sign it, but I'm not sure. That will put tighter restrictions on how much money can be paid to women. But that may have some implications on how quickly the research goes forward. On the other hand, you don't want women going through this somewhat risky procedure. I don't want to overstate the risks. 30,000 women a year go through it for fertility purposes. So they're competent adults making a decision that it's in their best interest, but it is a risky procedure. You wouldn't want poor women being exploited to get the money, just as you wouldn't want poor people selling their kidneys, though that's an even riskier procedure. So there are conflicting interests here. We have to figure out how to deal with them. There are, and this will only appeal if there are any other lawyers in the House, I think, there are intellectual property issues, patent issues. You may not have thought about Julie's bags here, Julie's beach balls being patented, but in fact they are. There are patents in the US on human embryonic stem cells. They're owned by the University of Wisconsin, and they're fairly stringent. There's an ethical question about this. Some countries have said you can't patent human embryonic stem cells. The European Patent Organization has said it's unethical, we won't allow it. Canada has taken 2 years and still hasn't decided what they think about it. The US said it's okay. So there's an ethical question. There's also a practical question. University of Wisconsin wants to hold up the California Institute for Regenerative Medicine for several $100 million for a license to use their patents. Do the taxpayers of California want to subsidize the cheeseheads? I'm sorry, the Wisconsin folks? To that extent, probably not. So we get into practical, legal, and ethical questions about intellectual property. There are some weird questions about interspecies issues. So human eggs are hard to get, but cow eggs are easy to get. Rabbit eggs are easy to get. There has been some research trying to make human embryonic stem cells by taking a cow egg, taking the cow nucleus out, putting a human nucleus in, and then fertilizing it. And that research is promising. It hasn't clearly worked yet, but it hasn't clearly not worked. Nobody wants to implant that and make a cow human or a human with long ears and a fuzzy tail. But it is, I think, fair to say a little disconcerting to think about that. Our Stanford colleague, Irv Weissman, has hypothesized an experiment that would take a mouse and put human stem cells in it, and this would be a breed of mouse that has the neurons in its brain die out at a certain age, put the human stem cells in, hoping that they would become neurons in the mouse, and that you get a living mouse, all of whose neurons are made up of human cells. Why would he do that? Well, not to have the mouse stand up and say, "Hi, I'm Mickey." Copyright violation to the Disney company, but forget that. So that he could have an in vivo, a living model organism in which to study human embryonic stem cells, human brain neurons. Because the best place to study them is in humans, but humans are really bad lab animals. You know, we don't follow directions, we're expensive, we sue, you can't slice us and dice us easily. So, but a mouse with human neurons also sounds a little unsettling. Well, more than unsettling, Senator Sam Brownback from the Theocracy of Kansas has proposed a legislation in the US Senate to make it criminal to either make part human, part non-human embryo-like constructs to make stem cells from them, or to make a non-human animal that has a brain that's substantially made up of human neurons. Irv could go to jail, which would be a great loss, especially since he's my next-door neighbor and a wonderful guy. And he grows great tomatoes. So last of these ethical issues is the most boring of all, but maybe the most important. I call them boundary issues or border issues. Only an accountant or a lawyer could love these, but they're really important. President Bush has said you can't spend federal money on research with human embryonic stem cell lines except these particular lines. Well, that involves drawing some borders. What does it mean to say stem cell lines, and what does it mean to say federal money? If you take a human embryonic stem cell and you successfully turn it into a beta pancreatic cell, a cell that makes insulin, can you use federal money on that? Because it's not an embryonic stem cell anymore. If you take embryonic stem cells and you take out an interesting protein from them, or some fluid from the group of them, can you do that? You're not doing the research with the stem cells, but it's something from the stem cells. I write ethics and law papers. Could I, with federal money, write a paper about the ethics of using non-federally approved stem cells? Or would that be using something derived from the stem cells with federal money and could land me and Stanford in trouble? That's unclear. It's also unclear what it means by using federal money. It's pretty clear you can't get a grant from the NIH. Okay, fine. But can you use a microscope that was bought with federal money? Can you use a postdoc who is supported 50% by a federal grant? Can you use a laptop that was purchased with federal money to write up the results of the experiment? Can you use the Stanford telephone system to make a phone call, which is supported in part by the overhead charges that the federal government pays Stanford for its share of the research? And so far, the answer to those largely seems to be, we don't know. But we worry about it a lot. So that's the bureaucratic part of me. And, you know, it's very dull, but it scares us. It could get us in a lot of trouble. We need to worry about it. And the result is people like Julie face incredible bureaucracy because we're trying to do what's right in the face of ambiguous and unclear rules. And that slows down the research. So, what's in the dish? Other ethical issues. Closing, some thoughts about science and society. Scientists don't like being part of politics, and I don't blame them. I think Bismarck was right when he said you never want to watch sausage or laws being made. It's very unsettling. But science is part of politics, for better or for worse. It's always been clear on the funding side. Who gets funded, whether it's the space shuttle or flights to Pluto, whether it's a planet or not, depends on politics. Whether the superconducting super collider gets funded depends on politics, or the International Space Station, or the Human Genome Project. We try to insulate some of those decisions through the careful process the NIH uses to try to select good grant applications, the one that the California Institute is trying to basically copy to keep politics out of the direct funding decision. But there's some mega politics involved in what areas get funded to begin with. But the stem cell fight has gone beyond that. It looks more like fights over, say, the Endangered Species Act or fights over global warming, but stronger than any of those. It's science in the middle of politics. It's become a partisan issue. It's true there are some Republicans who support the research, certainly a non-trivial number. There may be some Democrats who oppose it. But by and large, the two parties have different positions on this. And when we have Prop 71 and a California initiative and television ads one way or the other, we have this issue strongly in the middle of politics. And that's unfortunate. Because politics doesn't like arguments like, well, it's more likely than not that this will be good, but we can't be sure. Can't be sure is not a good political argument. Politics is about, this is evil, this is wonderful. I'm wonderful, he's evil. They're going to ruin the world, we're going to save it. Politics doesn't like subtle arguments. Science is full of uncertainty and subtle arguments. And so when science gets into politics, it makes people who like science cringe often because there are exaggerations in both directions. People saying that embryonic stem cells will cure things tomorrow. People saying that adult stem cells can do everything embryonic stem cells can, better, faster, and cause you to lose weight too. And that happens, and one reaction to it is that scientists, seeing this ugly process, tend to withdraw from it. And that's a shame because then we lose the expertise. It's really important that people like Dr. Weissman and Dr. Pizzo and others stay in the political fight one way or the other to make sure that good science is brought in. But what is most important, I think, in a democracy, decisions, science is part of society, it's ruled by society, it should be ruled by society. Our society is a democratic society. Winston Churchill said, I think quite accurately, democracy is the worst form of government, except every other type tried from time to time by the mind of man. Democracy is not an efficient, perfect form of government. It's ugly and messy, and happily we usually manage to muddle through. But we muddle through better when the citizens know what the fight is about. And so I'd like to end by thanking you for taking time out of the last day of August, of the end of summer, just before the Labor Day weekend, to come and hear about this, whatever your position is, whether you're in favor or opposed or somewhere in between, because this will only work well for us. The more citizens are educated, the more citizens care and are involved and engaged. Stanford has some ways for you to do it. My Stanford colleague Chris Scott is teaching a continuing studies course on Mondays this fall. That would be a great way to learn more about it. You can do Stanford on iPod. We've got lots of opportunities. Please take them. But thank you so much for being here tonight with us.
Well, thank you very much for that very provocative and important presentation. I think it's hopefully safe to say that in the last hour we've really gone through the broad spectrum of the science of stem cells, what they mean, how they work, what their promise is, and some of the ethical and legal challenges that surround them. Now, I think it's also pretty clear that it's getting dark and a little bit cold. And just think of it this way. If we don't do the stem cell research, we're going to be in a dark age anyway. So hopefully the next period that we have will provide some enlightenment. So feel free. We know that the questions are important, and anything you ask will be one that we would love to hear about.
Cord blood, stem cells from cord blood would be more like the embryonic stem cells and be more effective and more useful than the adult cells, the adult stem cells.
Dr. Baker?
So what we know about the cord blood cells is most of the potential there is like the blood stem cells, and that those are not as what we call pluripotent, that they're not going to be able to give rise to the neurons, the liver cells, the lungs. So they're much more pigeonholed into one category. They're not as what we call pluripotent.
So I just offer one caveat to that, and that is that in the clinic, we do use cord blood cells to replace bone marrow that has been destroyed. So actually, for what's called a bone marrow or a stem cell transplantation that might be used as part of cancer therapy or for damaged bone marrow, umbilical cord cells and stem cells that may come from the bone marrow both work, but that is very different from that therapeutic point of view from the use of those cells to create different types of non-blood forming cell populations. Who's got the microphone and who's going to pass it around?
I just have a quick question for you.
Okay.
If you, here at Stanford, because of the ethical dilemmas you guys have about the federal funding, are you now having separate labs for these things?
Oh boy, I can take that one away.
Don't take it too far.
Maybe to a Rastradero?
So right now, yes. So right now, all non-federal line human embryonic stem cell work is done in a completely separate laboratory than we usually. So I myself have two distinct separate laboratories that run pretty much separately on different money. How that's going to progress at Stanford, it's still not entirely clear.
Yeah.
I think what's going to happen is more and more people are going to start doing this work, and we're going to have to figure out how we're going to deal with that. But that's more DNP zone.
It's actually a really serious and practical issue. I mean, we simply don't have the space right now to create a new dedicated facility, nor do we have the funding for it at this point. In fact, our efforts to date in embryonic stem cell research are really because we had some donations from an anonymous donor that helped us get started. But you know, that's not enough to keep it going. It will be true that once the funding comes from the California Institute of Regenerative Medicine, that will provide a help. But as Professor Greely already told you, there's so much admixture of our resources that to create a whole separate facility just for this purpose is really not the wisest way to go, but right now it may be the only way we can.
Yeah, I don't think any of our labs have gone as far as one that Irv Weissman talks about, where the researcher has approved lines on one side and non-federally registered lines on the other, and has black and yellow police tape down the middle of the lab, just to make sure everybody remembers which side they're on and not to move a vial from one side of the lab to another side. It gets that ridiculous.
Yeah, but the problem is they're all breathing the same air that came out of the filtration system, and their only choice is really to follow the Brownback rule and be killed. All right, so we have a question back here. Yes.
Am I on?
You are on.
Gets pregnant and aborts very quickly. Could you get any stem cells out of what comes out?
Yes.
Yeah, so the short answer is no. So the time at which you would know that you did have a miscarriage is long after the time when we can actually harvest these cells from an embryo.
Right. And
after.
Right, and actually along that line, it's important to differentiate, because when that happens and there's a miscarriage, it means that those cells have already been implanted. In essence, a placenta has formed and the embryo is developing toward a fetus. And that is a very different process than the cells that you heard from Dr. Baker, which are just a small number of cells. And those happen, as you heard, within days. And there's no way that can really be discerned.
It is the case you can get some stem cells from aborted fetuses or miscarriages, but those are stem cells that have started down a line.
Right. They're not embryonic.
So you can get
cells.
neural stem cells that might make brain cells, but they won't make pancreas cells and skin cells and so on. They're not the basic pluripotent, the type that can make everything.
But they are differentiated, so you could use them, could you not, as regenerative medicine. And why is there no mention tonight or in the debate about 30 years of work in the Ukraine and China on fetal stem cells?
Well, there's actually a lot of work being done in the U.S., and Irv Weissman, whom I've mentioned several times tonight, works on fetal stem cells. But the problem, again, and Irv works on fetal brain stem cells. He thinks, he hasn't proven it yet, but he thinks he can use those therapeutically for brain cells. But he can't use them to make pancreas cells or to make skin cells or liver cells or other cells. So there are some valuable uses. Right. And if we could find 100 different fetal stem cells that could make each of the cell types, that might be the right way to go.
Yeah.
But we're nowhere near that at this point. And we do know that the embryonic stem cells, I come back to this, it's really important. We know they can make every cell type because we're here. because
Right.
they did.
Right. But I think one point that you made earlier, Hank, and Julie, you did as well, is none of us want to look at this in a mutually exclusive way. This is not about saying that we're only interested in embryonic stem cells. We are interested in the whole range of cells, whether it's fetal cells or adult cells or umbilical cord cells or embryonic cells. At the same time, we don't want one of them to be excluded because that may be the route to the answers that we're all seeking.
Fat cells. I'm looking for those stem cells from fat cells. I can become
Well,
a
we do-
natural resource.
Well, we do have actually an investigator here, Michael Longacre. If he were talking to you tonight, he would tell you that he could, in his laboratory, actually harvest stem cells from fat. And if the way America's going, this will be a growth industry.
And so that's why Mike has that ugly look in his eyes when we talk.
Indeed. He comes from the Midwest, where he says this is more common. Okay.
Yes, my question concerns the 400,000 frozen embryos now in the IVF clinics, and how long practically those would last if it were completely legal and possible to use them. There was a RAND Corporation study done in 2003 that found that 86% of those were designated for future use by the donors or by other people, and they found that I think it was 2.6% could be used for stem cell lines, and that would increase the number of existing stem cells available for federal research if the feds approved it by a factor of about 10. But the indication I saw was that still that wouldn't be enough. So is it realistic that even if all this were legalized, that we would have to move after a certain time, and how soon, to the buying and selling of fertilized eggs?
Well, first, it's not illegal, except in a couple of states. So it is legal in most places. But you're right, one would have to go back and reconsent the parents who had created those embryos, for most of them, because most of them were not asked and did not agree to let them be used for research. California, about 4 years ago, passed a bill requiring that in vitro fertilization patients be asked whether they want leftover embryos to be used for research, but California is unusual in that. How many of those folks would agree to the research isn't clear, but we do know that an awful lot of those embryos never get reused. The couple that froze them has been successful and doesn't want any more children, or has given up on the idea of having children. So there are IVF clinics that have 10, 15, 20-year-old frozen embryos that they don't know what to do with. No one's ever going to try to implant them. Nobody would dare to try to implant one that old for fear of what the possible health consequences might be for the child. And you know, in more recent years, the IVF clinics have gotten smarter and charge an annual fee to keep them frozen. And that ultimately forces parents to decide, or forces the patients to decide, whether they really want to keep this option going. So you're right, they couldn't all be used now. You'd have to get consent, but you may well be able to get that consent from many of those parents.
Yes, do we have a microphone? Where's our microphone?
We've got lots of hands.
Whoever has the mic.
I got it. Is United States using scientists to rest of the planet where there are more efficient, effective democracies?
That's for you, Phil.
Say that again? The
Are we
question?
losing scientists to other
Yes.
places?
Yeah, the answer is yes. And a couple of things about that. First of all, over the last 2 to 3 years, we've actually been able to attract some wonderful scientists to join us here at Stanford because they were interested in being able to do this work. We're now having a much harder time, and in this last handful of months, we actually lost 2 investigators that were going to come here to Korea, and the reason to Singapore, and the reason they went to Singapore was because it was a much more permissive environment, and they could carry out their work. The University of California at San Francisco, a handful of years ago, lost one of its most premier scientists to the UK. And so we will see that migration happen. You know, we're all sitting here in Silicon Valley, and you recognize that Silicon Valley is the beneficiary of information technology and, to an increasing degree, biotechnology. That can all change. If we don't have stem cell research here and someone else develops it, we'll lose not only the scientific opportunities, but we'll also lose the economic opportunities as well.
Yeah, the scientists are the most valuable assets, and when there are fights going on within the country, is this a problem? It's a major risk for the country, or?
I think it is an absolute risk for the country. There are people who are absolutely passionate and committed to this work, and they will leave to do the work if they cannot do it here.
There's a little bit of data on this from my colleague at the Biomedical Ethics Center, Jen McCormick, who published a paper in Nature Biotechnology showing that I think it was 5 years ago, 70% of embryonic stem cell papers published, peer-reviewed papers published, had an American-based first author. 2 years ago, the most recent data she had, it was 32%. Right. Now, small numbers, small n, but it's not a good sign.
Right. And I think there's one other factor, and Julie may want to comment on this as well, which is one of our most important opportunities is to train young people in various scientific areas. That's where the best ideas come from, quite honestly, and certainly where the work gets carried out. And our pipeline of young people, smart, energetic young people pursuing stem cell research has really dwindled because no one sees the opportunity ahead. Now, honestly, we have, I think for the first time, a little bit of hope in that regard because of California's Stem Cell Institute, which recently funded and is now supporting some trainees. That pipeline is critical to our future. Shut that off and we lose a generation. Julie, do you want to add to that?
I just agree, yeah.
The 16 cells that are the embryo, are they all stem cells?
That's a good question. From what we know, they are. So that's all I can say. Yeah, we think they are all stem cells, but it's very difficult to actually tear apart those 16 cells and analyze them individually to ask, genetic questions about what those individual cells are. So from what we know, we think that they are.
But we aren't sure.
But we're not sure.
And the ACT research I talked about earlier, they took 12, 15 embryos at the 8-cell stage and completely broke them up. So they got 150. And they had 100 different individual cells made from breaking up a bunch of different embryos. 2 of those turned into cell lines. Now, maybe it's just an inherently inefficient process. Maybe some of those cells in the 8-cell embryo already could not become an embryonic stem cell line. Mm. We don't know yet, but that's a great topic for a lot of research.
Right. But there is one other piece of data which is an interesting one, because the technology that you described has been around for at least a handful of years, and it's been known that at the 8-cell stage you can actually take out one of those cells. And yet the remaining 7 will go on to produce a human being, which is fascinating. If you get to the 16-cell stage and you take one out, you have a problem, right? So there is a critical point in time. And in fact, the reason why that technology began to develop is the possibility that at the 8-cell stage, you could actually take out one of those cells and do a full battery of genomic and genetic testing to determine whether or not there's some genetic problem in the remaining 7 cells that might lead you to choose whether or not to implant that particular 7-cell embryo. So this is an intriguing area, and we're going to see lots more of these, fascinating both issues and dilemmas emerge in the years ahead. Okay, where do we have our next question?
Hi, right here. In the middle.
In the middle.
Right here.
Okay, wherever you are, speak.
Right here.
We can hear you, but we can't see
dancing.
you.
Oh, there you are.
Down.
There you are. We need.
Hi.
Great, thank you.
My question has to do with maybe teenagers who want to have an abortion. At that point in time, is it too late to take any type of stem cells from the fetus?
Yeah. I think we've already partly addressed that before, and I think the answer to that is yes. Julie, do you agree?
Yeah, it's way too late.
For the embryonic
stem
cells.
Right. We're talking 5 days. After 5 days,
right.
There's been no report of derivation of human embryonic stem cells from anything after 5 days, which is way before most people know that they're pregnant.
Right. Most people are not going to know they're pregnant till close to a month.
Right. It hasn't even implanted at that point.
Right. Okay, where's our next? Yes, here we are right in the front.
I have a question about the IVF patients and what type of relationship you're developing with them to encourage their participation in the supply chain of the cells, and what kind of ethical and moral issues there are for you to encourage a choice of donation rather than discarding embryos?
That's a great question.
Let me take that if I can.
Yeah, go for it.
Actually, Julie should not be developing a relationship with them. The goal, and there's some very influential guidelines put out by the National Academy of Sciences and Institute of Medicine in April of 2005. Those guidelines, which are generally viewed as being a good job, say that the person who gets the informed consent for the research use of embryos, sperm, eggs, and so on cannot be one of the researchers intending to use it. You don't want Julie trying to convince, or we worry a little that Julie will be too persuasive. We don't want her trying to convince a woman who's making this decision to definitely do it. We want her doctor, her IVF doctor, who's got a relationship with her and a doctor-patient relationship with her, to be the one who raises that issue, gives the information, and either gets the consent or not. So, could we get more eggs and embryos if we turn Julie loose? I think so. But I think it's probably cleaner, safer, less of a conflict of interest this way.
Right. And actually, one of the things that brought down initially the whole Korean group was that very fact, that the investigator who was pioneering at that time, seemed to be pioneering this work, was in a sense coercing people in his own laboratory to provide eggs. So I think it is really critical to have these separations. Now, IVF physicians who are providing care to her or his patients certainly can speak with patients about their views about how they'd handle the embryos in the future. And that dialogue, I think, is perfectly appropriate. Yes, Frank.
Question for Julie. You mentioned that some of the embryos are maybe diseased, you can tell. How can you tell that a particular problem exists? And if indeed there's a problem, I assume that the donor would not want to use that. In which case, what are the ethical problems with trying to use that in your lab? And for Dr. Piso, could you possibly open a campus or a lab in the country that allows this and get the monies from them? What would be the legal problem with that?
To open a campus where?
Singapore.
In Singapore. You'd like me in Singapore, okay. There are a lot of people that would like me in Singapore. But yes, Julie, please.
Okay, I'll take the first part. So there's something called pre-implantation diagnosis that the IVF centers do. So that basically is, for example, a couple comes in that has 2 children that are already affected with something like cystic fibrosis, and they want to have another child that isn't affected. And so they come in and go through the normal IVF cycle, even though they could have the child, naturally by themselves. They come into IVF and go through a normal IVF cycle. The IVF team gets the embryos, and then they actually take one of the cells off at a very early 4-8 cell stage, and it's sent to a laboratory where it's examined for whether or not that cell carries the mutation that will lead to that disease. If it does carry that mutation, then clearly the patients, the couple, doesn't want those embryos, and those embryos are generally discarded. Right. And those are the ones that we really, really think are incredibly valuable, and they are part of the discard pile that we certainly would obtain.
And there is one other technology that we would love to be able to use as well, called nuclear transfer, that can help with this as well. And what that means is that you can take the nucleus from an adult cell, from a skin biopsy as a case in point. So if someone has Parkinson's disease or Alzheimer's disease or Lou Gehrig's disease or cancer or diabetes, if you take a skin cell and in the laboratory remove the nucleus and implant that nucleus in an embryonic cell or in an egg, you can create an embryonic stem cell that bears that disease. And that is a incredibly powerful tool, potentially. We can do it in mice, and there are investigators that know that they can do it in humans as well. That that tool really can be transformative, because that would allow us to develop a whole cohort of disease-bearing cells, people with cancer, where we cannot study all of the genetic abnormalities in the kind of time sequence that this would allow us to do. And so we're hoping that nuclear transfer will complement some of these other technologies that Dr. Baker has talked about.
And that's patient-specific.
Yes.
So it would be your own cell, so you wouldn't have to worry about immune rejection or anything like that.
Right. And to your question about moving Stanford elsewhere, I don't know the answer as to how that would be viewed by those who oversee the university, but I do feel that two things. One of them is that we are very concerned at Stanford about doing everything as properly and as ethical and as well-reasoned as we can, because we recognize that we're very dependent as an institution on the funding support that we get from organizations like the National Institutes of Health. And, as Professor Greely said, we live in a very political world, and that world is not without some degrees of recrimination. And so were we to do something that looks spiteful, it might have other unintended consequences that would unfold as well. So that's why we're happy to be here in California.
One of many reasons.
However, if we were a little bit north of here, it would still be light at this time, and that might help, at least for a little while longer. Okay, I think we're beginning to run out of time. We'll take just a couple more questions. Yes, one here.
So you have 8 cells in the Petri dish. How long before it turns into 16, and what's the ultimate limit it will end up in the Petri dish, and how do you stop it at 8 so you can do your research on it?
So I'll answer one of the questions first. So the ultimate is shortly after this big balloon over here. Shortly after that time, it's going to do something called hatching, where the outside breaks open, and then it needs to touch the uterus to have exchange occur between the mother and the. So it's a day after this stage where it needs to be implanted. What was the?
How long do you have to work with it? In other words,
So it's
only hours or days, and then all of a sudden it's out of.
At the 8-cell stage, it goes to 16 in about 12 hours. So from a fertilized egg to the blastula stage, which is what this is about 5 days. So it's in culture for about 5 days.
And you have to do all your research at that point.
No, then you've got the cell line.
No, then we have the cell line. So this then becomes a cell line.
So how do you preserve the cell line? Does it keep multiplying, or what?
Yeah, so that's one of the beauties of the human embryonic stem cell, is that it's a stem cell, and one of the properties of stem cell is it self-renews, and so you can keep that going in culture indefinitely.
But doesn't it turn into a human? I mean, in other words, isn't it trying to turn into a human at that point?
It stays as a stem cell, and the only way that we get it to become neurons and et cetera, all these different cell types, is by subjecting it to different hormones, taking it off hormones, putting it on different matrices, exposing it to different environments. Otherwise, it's going to stay as a stem cell line, it won't become anything.
So that cell line that Julie talked about that's being used in most of the research, that's 6 or 7 years old. There are probably tons of cells from that cell line spread all around the world. They're staying as embryonic stem cells until we take them out and make them into something else. They won't become babies.
Right. So take one. Does someone else have a microphone? We'll take
Yes,
one last question.
I do. In the ethical discussion, do you ever examine the dogma of ensoulment, that the sperm and an egg get together, and at that moment, one soul arrives? There are logical problems with that. Dr. Pizzo talked about the implanting of a nucleus into a cell. Well, there's no sperm and egg coming together there. Why do we call that an embryo at that point? Or if 1 out of 8 of us were originally part of a twin pair, as I heard in a documentary the other night, but only a few of us get born and the other half dies, are 1/8 of us half-souled? Or have we stolen the soul from the other? Or we zombies?
Those are good questions for the people who believe in ensoulment at the time of fertilization. And they're smart people, and they've got some answers for them. They're not, to my mind, particularly convincing answers. But it's hard to answer that from a scientific basis because we can't detect a soul. We don't know where it is, how to find it. We don't know what it looks like. We don't know whether it exists. So we can't give you a scientific answer to when ensoulment occurs, if it ever occurs. That's a position people take based on faith. And that's part of what makes it so difficult to reach agreement. It's hard to imagine experiments we could do that could convince people who hold one view to shift to the other one. So all I can tell you is there are a lot of smart people who work on this issue from lots of perspectives. There are a lot of smart people in the Vatican and elsewhere. They have answers for those questions, but they're not answers that I at least find very convincing.
Right. So I think on that note, we'll thank our speakers. And I would say there are also a lot of not so smart people who have views as well, but that's a different story. And I hope tonight you've had an opportunity to really both engage in the content, learn something about this topic. Obviously, you can see there are many more questions than there are answers. I hope you'll keep talking about it, that you'll talk with your family and your friends and your neighbors, that you'll learn more about it. If you go to our Stanford Medicine homepage and plug in stem cells, you get to a whole series of fact sheets and questions and answers. We'd love you to become more informed, to become better advocates for what we're trying to do, both in this area of science and medicine and every other. And we thank you very much for coming tonight, and be careful as you leave because it is now dark. Thank you,
The preceding program is copyrighted by Stanford University. Please visit us at stanford.edu.